আশা করছি লাইভে চলে এসেছি। লেটস চেক ওয়ান্স। ইয়া। ওকে বন্ধুরা, চলে এসেছি এই সময় লাইভ ক্লাসে। তো এই লাইভ ক্লাসে আজকে যেটা চেষ্টা করব, কিছুটা ব্রিফ ডেসক্রিপশন অ্যাবাউট দ্য early development of C. elegans, normally in the genetic study and also developmental study. This organism is very much important. So আজকে আমি মোটামুটি উম, ভেবেছি যে এটা নিয়ে শুরু করব, কিন্তু এখন বাজে ঘড়িতে 12:05। তো হয়তো পুরোটা হবে না, কিছুটা পোর্শন ডিসকাস করব। তো তারপরে আবার নেক্সট কোনো একটা দিন যদি ওটাকে ডিসকাস করে নেওয়া যায়, মানে পুরোটাকে শেষ করা যায়, পুরোটা বলতে একটা সামগ্রিক অ্যানালাইসিস, নট কমপ্লিট এন্টায়ার অ্যানালাইসিস। তো সেক্ষেত্রে কারণ এই জিনিসটা অনেক বেশি বড়, তো আহ, চেষ্টা করছি যে আহ, পুরোটাকে কভার করার। হয়তো প্রথম দিকে হয়তো হবে না, আহ, কিন্তু আশা করা যায় যে এটা ধীরে ধীরে আমি সি এলিগেন্সের যে ডেভেলপমেন্ট বা আমাদের যে ডেভেলপমেন্টাল বায়োলজির যে সেশনটা আমি অনলাইনে বলার চেষ্টা করছি, যেখান থেকে হয়তো অনেকে আহ, কিছুটা জানতে পারে, হয়তো যারা নন ব্যাকগ্রাউন্ডের আছে তারাও এই বিষয়টাকে অনেক বেশি আহ, বুঝতে পারে। লাইভে দেখতে পাচ্ছি চলে এসছে কেউ না কেউ, আর না হলেও পরেই লাইভটা দেখতে পাবে লোকে। তো আহ, আশা করা যায় 1 মাসের মতো থাকে, কিছুজন হলেও দেখতে পাবে, এবং যেখান থেকে ডেভেলপমেন্টাল বায়োলজি কিভাবে আহ, ধীরে ধীরে মানে এগিয়েছে তার সায়েন্সটাকে বুঝতে পারবে। তো আজকে আবার এই ক্লাসে চলে এসেছি। তো আমি এবারে ভিডিওটাকে ছোট করে নিই, ছোট করে নিয়ে ক্লাসের মধ্যে ঢুকে পড়বো। আশা করছি তোমরা দেখতে পাচ্ছ স্ক্রিন, যেটা আজকের মেক ইট মোর স্মল, দ্যাট উইল বি বেটার। ইয়া। সো এই ক্লাসে যেটা আজকের মেজর ডিসকাস হবে সেটা হচ্ছে সি, মানে আমি যদি পুরোটা লিখে দিচ্ছি, যেখানে তোমাদের আহ, এই নামটার সঙ্গে হয়তো অনেকেই পরিচিত, আবার অনেকেই আহ, পরিচিত নয়, মানে যারা নতুন নতুন হয়তো বায়োলজিকে ভালোবাসছে। তো কারণ এই মাইক্রো অর্গানিজমটা একটা মডেল অর্গানিজম, তো মডেল অর্গানিজম হওয়ার কারণে আহ, এটা নিয়ে অনেক বেশি কাজ হয়েছে। তো সি এলিগেন্স, আর মেনলি আজকে আমরা তা দেখছি ডেভেলপমেন্ট। তো নর্মালি যে সি এলিগ্যান্স সেটা হচ্ছে একটা নিমাটোড। তো ক্যানর হ্যাবিটিস এলিগ্যান্স যেটা হচ্ছে একটা নিমাটোড। তো ওই নিমাটোডটাতে প্রিসাইজলি ইউ ক্যান সে অ্যাবাউট দ্য 5959 somatic cells are there, and also like they have their lineage through a cuticle. So this is the invariant type of that. The that means like I can write it, that's the invariant type. That means in invariant, it means meaning each cell each cell consistently each cell consistently give rises rise to the same number. And type of cells. So that means you can see these are the somatic cells, and every cells rise to the same number, like 959. Let's talk about we already we have studied the cleavage portion, so we have just remind that this is the type of the rotational cleavage. When Okay, let's see. Okay, in this is the rotational cleavage. I think that mode is good for me. So this is the cleavage portion, like cleavage is the rotational cleavage. So this is the rotational cleavage is there. So that means like you know about the in mammals there is also the rotational cleavage. So in zygote portions we already know there is a some anterior part will be there and some posterior part will be there. So in the anterior and the posterior part, like let's say it's a Acha. And the another information you should remind about that. I think this pen is not good. Yes. Yes. So that is the holoplastic. That is the holoplastic cleavage. That means holoplastic cleavage, it means the complete cleavage. Complete complete cleavage. So in C. elegans, that is the in early development, there is the asymmetric cell divisions are there. So in early development, in early development, in early their asymmetric cell division. I think you can make some notes because in that I'm just making this kind of lecture for making some brief type of lecture. It's no kind of it's not a lecture. It's kind of notes taking session like that. So in that thing, that kind of you can say brief idea about that, and also this kind of exam type of thing. So C. elegans, it's a model organism, and there have 959 somatic cells. There's an invariant type. Each cell it continuously gives some rise to the same number, and cleavage, it's a rotational cleavage, and also it's a holoplastic cleavage. That means the complete cleavage. In early development, their asymmetric cell divisions are there. So now we are going to the zygote part. Let's say it's a zygote. So in zygote part, there are two type of things. One, this is a asymmetric division. So asymmetric, it means some part will be the anterior and some part will be the posterior. And already we know it's a asymmetric. So in anterior part, that is called the founder cell. In anterior part, the cell will be the founder cell. And in the posterior part, that is the P1 stem cell. It's a P1 stem cell. So kind of two type of cell. You first you have to know about the founder cell. Like that's called the progenitor cell. That's the founder cell, or you may write down it's a progenitor cells. So there is some restricted potentials are there. They give the differential potential on that. So that means it's just a condition specified. Already we have discussed the condition specified, and this require the stem cells near vicinity. And so this zygote, this is also a type of fluid stem cell. It's a pluripotent stem cell, multipotent. So it's a kind of stem. This is a stem cell. Now you got the founder cell and P1 stem cell. So this P1 stem cell have their more higher potency. Now we can write it the specifications of the founder cell. Number one, yes, that is the restricted potential, and number two, that is the conditional specification specified, and in P1 stem cell, that is the automated or automated or autonomous specification. So two type of specification. We already discussed the conditional specification and automated specification. So here we all see that the founder cells is the conditional specified, and the P1 stem cell, that is the autonomous specification. So that means, and also we have to remember that the fate cannot be changed. They can independently form all types of cells. Types of cells. So this is the portions whether we know about the cell lineages. Now the major important things is the cell lineages of the C elegans. So we have already discussed the basic thing. Now we have to remember the few cell types. Like, let's start with the zygote. So in that zygote, already that is a P0 stem cell, and then you already know two type of lineages already. You know that one is the founder cell, founder cell, founder cell, and another is the P1 stem cell. So the founder cell, that is you know about the AB, it is called the AB cells, and P1 you know the P1. So that the founder cell is the AB cells. So AB express the two type of cell lines. One is the AB ABA, and another is the ABP cell. So two type of cell line. So that is one is the ABA, one is the ABA, another is the ABP cell. Now you have to remember the ABA cell, that means the few gene expressions are there. So now ABA, from ABA, the two type of cell lines are there. A, B, A, L, another is A, B, A, R. So founder cell, ABA type of cell, and then another is ABP cell. I'll coming with the ABP. So now ABA type, that is the L, ABAL, ABAR cell. So this is the in ABA cell types. What are the ultimate form? That is the pharynx. Sorry, it's a mistake. It will be PHARYNX, pharynx, and the hypodermis and neurons. So these are developed from the ABAL cell, ABAL, ABAL. Now we are talking about the ABAR, the part of ABAR. So these two type of cell, like ABA and ABR, both the cells helps to develop the pharynx, hypodermis, and neurons. Now we talk about the ABP cells. These are also two type of cells, easily A, B, P, L, and ABAR. What are the major functions of that cell? The major functions of these, the neurons and the hypodermis. So neurons, we have right one. So these are the neurons cell. They are developing from the AVP cells, and these are the major type of the founder cell or AB cells. So the total number of cell here is the 389. So these are the 389 cells are from this the founder cell. Now we talk about the P1 stem cell. So from the P1 stem cell, two types of stem cells are seen. One is the MS cell. Let's talk about the EMS. EMS cell. What are the EMS cells? And from this EMS cell, two types of cell lines are developed in the C. elegans development. Another is the MS. What happened? Sorry. Yeah, some technical glitch. So another is the MS cells, and the another is the E cell lines. So from MS cells, from the MS cells, they are muscle and gonads, and these are the number is the 80. And E cell, from the E cell, it develops the intestine, and the number of the cells is 20. So now we get the 489 cells. So the total number of the cells, in previously I have written here, there are 959 cells. The total cells are there, and already we know about the 389 and 80 and the 20 cells. And we have seen that their muscles, gonads, intestines, these are all developed from the EMS cells, cell line cells. These are their precursor cells, and these are also coming from the P1 stem cell. Now we talk about the another part from this P1 stem cell. Another is called the P2 stem cell.
So from P2 stem cell, there is a two type of lines. One is the D line. Let's say one is the D line, and another line is called the from P2 two type of line. One is the C line and the P3 line. So from C line, it's developed the muscles, muscles and the two neurons. So it's about the 47 cells. From P3, there is two types of cells. Okay, so from P3, there is a two types of cells are there. One is called the D cell and another called the P4 cell. From the D cell, it is also responsible for the muscle development, 20 cell. And from P4 cell, there is a two type of cell line. One is called the G3, another is called the G2. So these two lines are the germ lines. Germ line, that is a two cell. So this is the complete development, and there is a pattern, cleavage pattern. Let's write it. Cleavage pattern of C. elegans. So now I make it summary from beginning. Uh, the founder cells undergoes their equatorial division. It means their longitudinal division. They are 90 degree to the anterior and posterior axis. Whether they are P cells, this is the P1 stem cells, like P1 cells undergoes their meridional divisions or transverse divisions and make it one of the founder cells, that is the EMS cells. So you can write it here. Already I have written in that the founder cells is the kind of conditional specified and these are autonomous specified. Okay, rotation I think we should write down. That will be in another color. Take the green. So the founder cell, it is needed the equatorial equatorial division, and this is P1 stem cell and it's a transverse division, and from the EMS cell and P2 from founder cell, that is the AB type of cells, and that divides two type of cells. One is the ABA, another is the ABP, and from the ABA they are ABAL and ABR. Their major functions to for the development of the pharynx, hypodermis and neurons. Though other type of cells like ABP, that also forms the two type of cell lines, ABPL and ABAR, and that forms the neurons. Whether they are P1 stem cells, these are transverse divisions. In that region, the EMS cells are in our form. This is also one of the type of the founder cell line, and these EMR cell lines demarks their ventral region of the developing embryo. So already in that EMS, that also works on the ventral region. That's for the reminder. So in that ventral region, there are also two type of cells. One another is the MS cell line, MS lines, and another is the E. MS is also responsible for the muscles and gonads, and it's all about their 80 cell lines. 80 cells are responsible for that, and also E line responsible for the intestine development for the C. elegans. And from P1 to P2 and from P3 and P3 to P4, these are responsible for the germline cells. So somewhere some kind of questions are there, like the jam lines are coming from. That's not directly from P2. That's also P line. They are P1 stem to P2, P2 to P3, P3 to P4, P4 to the Z3 and Z2. These two type of cell lines are the jam lines. Hope that they are half an hour they are gone. So in that half an hour we know about the some C elegans, different type of cell lines, and we know about the two type of these divisions. One is the equatorial division, another is the transverse division, and also we know about their potency, the different type of potency. We know about that there. From P0, it's their ultimatum potency, and then when it's from their founder cells and P1 stem cell, and in that specific region their potency is get restricted. So that means in that diagram you can see their entire page is shown, their potency is kind of decreasing when it is coming to the top to the bottom side. So yeah, you can see this arrow shows that the potency decrease from this the P0 to the P4. Hope that you can understand that some of these lines, like how these kinds of the cells are formed. And also we have seen that these are kind of first left right asymmetry appearing, and from their 4 to 8 scale of stages, and whether two of these granddaughters shift anteriorly during the formation. So how it's happening? Let's take another page. Yeah. So this is the, let's talk about, this is the let's see the zygote one. Another part is called anterior, another called part is called the posterior. So when it's coming to that two type of cell, already we know about that. In that zone, specific zone, we know about the AB. We can remind like A anterior, AB site, and another site is the P site. So there's a P1 stem cell. Let's, and these are kind of chorion. Let's say this region is the chorion region, and then you can see in that step, like this is the cell AB. Already we study about the two types of there. One is the ABA type and another is the ABP type. And this P1 cell, we know about that the one, and one is the EMS cell and another is the P line, like P2 line. So these are the chorion part. Let's make it green. I think illustration is not good, but fine, we can understand. Oh, this is the kind of EMS. Then this. Let's make it green. Single line. Yeah. So first this cell line anterior to posterior. Let's make it that. So anterior to posterior ABP, and then ABP, ABA, EMS P2. So then let's make it the straight line. Then it will be well understood. Like this way and this way. Then let's make it large. Then you can understand there are two type of cell line lines. Another is the ABA L and AB AB ABAL and ABAR, like ABAR. So their movement will be in that direction, and in let's say it's a ABP, ABP, R and ABAR. So their movement, I think ABL, sorry, I think I make it wrong. Okay, let's redraw. Then it will be like clearly understood. Let's say it's a C cell and C and P3 will be there in that state line. MS and E will be there in that state line. MS and E and AB, AR, sorry, ABPR and ABPL will be in that state line, and ABAR and ABAL will be in that state line. Yes, now make it more clear. So that type of rotation happen. I think this cell will be like intact with each other. For the diagram purpose, it's not a good one, but still you can understand these are the types of cells in the C. elegans development. Now the two cells when it's developed it, there are two cells like this is the, let's say this is the diagram of C. elegans. So germ cells are found in that region, the germ cell. So I think you guys can understand the types of the cells in that C. elegans development. So and few information about that. The sperms are stored in that way. The mature egg must pass through on the way to the vulva, and also germ cells near the distal tip of the gonad undergo the mitosis. So I think these are the types of the cells and different sequence. Now we are going to the fate of these determinations, like P1 to AB founder cells, and also the form of these all cells, and form their tractions of cells like AB to P1, ABAL to ABAR. So these are kind of fate. I have already discussed their fate and the cleavage pattern in that diagram. Hope that you guys can understand about the different types. Now their control of the blastomeyer activity. So I think today uh, time is 12:37. Okay, fine. Fine. So we will discuss some of the genes. Those are the important genes, but for that you have to remember this one already we have discussed in 37 minutes or 32 minutes. So let's talk about some genes, and that gene you can see these are the genes, kind of the morphogenes. We talk about the some morphogenes, kind of their morphogenic determinant or some of the signal present in the cytosol. Let's say it's a cell or zygote. So in that zygote, few morphogens like some of the signals are there, and these are the determinant factor. So what are the determinant factor here? One is the skn1, pal1, and the pie1. So these three are the transcription factor, you can say, or you can say the signal within the cytosol. These are already present in the zygote. So they are controlling their morphogenic changes. So that means they are the morphogenic determinant. Let's say about the SKN1. Already we know about the EMS development or EMS cell. So this SK1 determined the EMS cell fate. Like we can write here EMS cell fate.
Now we talk about the PAL1. So in PAL1, in PAL1, their determination of the P1 stem cell, and also we talk about the pi1. Pi1 is mainly these are responsible, or that can help the development of the germline. So these morphogenetic determinant, those are present in that zygote. Now we talk about the EMS cell determination. So like for EMS cell, some pathways are there. One of the key pathway is the WNT pathway. And how SKN1 works. Like already we know about the SKN1. There are transcription factor or morphogenic determinant that are already present uh, in the cell. So that help the two type of thing. One is the MED1, another is the MED2, and that help that the E and MS cells differentiation. And if they are, one of the important factor is the POP1. If POP1 stimulate, then MS cells develop and going to the pharynx and muscle development. If so, like I'll go through the pathway in the different day, because otherwise it's kind of messy and heavy one. So today, for today, the WNT pathway, fine. I'll just write down that pathway, and next day when I take the classes, I'll go for the complete details. Like let's say for today, I'll just write down the pathway. That is the SKN1. SKN1. So for development, it's trying to develop the EMS cell, and it's in the MED1, MED2. Then it's EMS. Now the two important key factor, another one is the POP1, POP1 and the TBX 35. Now POP1 stimulate and form the MS, and MS2 pharynx and muscle. So these are the developmental step of EMS cell fate. Like that, some another factor we already know, PAL, and that PAL to the C fate or hypodermis, muscle hypodermis space. In that factor, the TBS is inhibit that. If it is inhibit that, then this fate will come. And for P2 stem cells removed by the fourth stage, and then they are like MS2, MS cell, and E cell and MS cell. So these are the pathway, brief pathway. So from this class, we have just make it summarize from that. We have already know about the C. elegans. It's a model organism for the developmental study. So in that developmental study, there's fixed number of somatic cells are there. So like 5959 type of cells, and they are invariant asymmetric divisions, and these are rotational cleavage. The already we know about that. Also mammals are mammals cleavage is rotational cleavage, and also C. elegans cleavage is rotational cleavage, and this is the holoblastic cleavage, and this is a complete cleavage. So in early development, already we know about some of the anterior part and some of the posterior part, in the anterior part, their cells is the founder cell, and the posterior part it's a P cell. So it, from the zygote, the founder cells is you can say it's a AB cells, and that can divide into the ABA and ABP, and ABA to ABAL and ABAR, and these are responsible for the development of the pharynx and hypodermis and neurons. And it's all about 389, the number you, if you calculate it. And the another type is the ABP cells, that is the ABPL and also ABAR cells. Those are responsible for the neuron development of that from founder cells. And these founder cells are the equatorial divisions found during cleavage. And from P1 stem cells, they are trans like the EMS cells and P2 stem cells. From EMS to MS cells and E cells, and MS, those are responsible for the gonad development and muscle development, and from the E cells, that is the intestine development. So from the EMS cell and from the P2 stem cell, they are C type of cells and the P3, and from P3 to the D type of cells and P4 cells, and these P4 cells are responsible for the two type of Z3 and Z3 and Z2 cells, and these are the germline 2 cells. So these are the kind of cells, and these are cleavage pattern of the C. elegans. I think in that complete diagram you can take some screenshot over there and make it more easy and make some notes about that. And then talk about some of these changes to in the diagram, and also we said that some of the morphogenic determinant, one is the skn1 and the pl1 and the pi1, and this skn1, that is the EMS self-fed determinant morphogen determinant, that means that can stimulate the changes for that EMS cell division. And the another is the PL1, that's the P1 stem cell, and another is the PIE1. This is the germline division. Hope that this is the today I want to make it off, and then the next class I will discuss about the some PR proteins and some of these like anterior posterior axis determinant, and also like ABA and ABP in the if they are in a normal position in the embryo, and something happening in this position, you if you change some of this position, what can be happen there. So like some of these notch pathway and GLP-1 pathways, and along with some of the Wnt pathways, how they are involved on that specific process, like POP1 deficiency, what happened their EMS2, that no such kind of MS cell will form if their POP1 deficiencies are there. So these are things I will discuss in the next class, and then we'll go for the sea urchin development, like uh, first the C. elegans, and then we'll go for the sea urchin development. So these are the things I want to discuss with that live, and hope that I have covered it, most important things on that. So you guys can understand that, and you can make some questions if any doubts are there. Kind of these are kind of very basics. Hope that in that zone, those are not from that background. They might have some questions, but those are from already biology background and already studied with that. They can just revise it with seeing that video. And these are not too complicated. If you want the more complicated or more difficult class, and relatively expression changes, and how research is going on with that elegans research, or this kind of model organism, and how the gene changes, and if you want to stop some genes, how the expression changes, and some cells will not develop, and what are the key factor and major factor. If you want to know about more details, you can also message me, and if I get time with that time zone, I can discuss with you, and in a major time I cannot give you. So but you can, if you need that, and then I can you can discuss with you. Hope that this live can uh, enlighten you about the C elegans development if you are not from that background. And if you know about that, then that will kind of revision. And if you are a student of class 12 or might be in the bachelor's, and then that is useful for you. And if you are master student, then it's kind of revision, and that's not needed. But no, all biology students are not from the developmental background. Like when I was a bachelor student, the developmental biology was there, but in another way, like eye development, kind of human parts development is there, not the these are like sea urchin development, sea elegans development, and wing development. These are not there in our curriculum, but we have learned it. So kind of different. Developmental biology is too much, and so in any biology syllabus all are not there if you are not exactly bachelor's in developmental biology, but still we have to learn it from exam purposes or some learning purposes. So hope that that video, if you see entirely, it can give you some information, and then you have to study. That's the thing. Good night. See you in the next video.